Welcome, everyone. Thank you for attending the Citizen Science event about Aqua Bugs, Invertebrates, and Water Quality. My name is Nicolette Michael, and I am an outreach assistant with the Center for Environmental Inquiry. Our public events are usually done at either our Osborne Preserve on Sonoma Mountain in Pengrove or our Galbraith Preserve near Yorkville in Mendocino. Okay. We are all feeling the challenges of shelter in place like you all, and we're happy to be able to reach people during this time and connect in new ways, like during this event. Before I let Wendy St. take it away, I want to tell you a little bit about what the Center for Environmental Inquiry is and how we can be a resource to you, no matter if you are affiliated with Sonoma State or not, whether you're a student or parent, government employee, educator, member of the public, or an organization in need of environmental solutions. The center envisions a North Bay working together to find sustainable solutions and invites all of you to get environmentally ready with us. We're building a community of learners and problem solvers across all sectors of society, and we're providing firsthand understanding of our connection with nature, and we're building skills to help solve sustainable solutions. We're helping people become more aware and prepared and engaged in our society as a whole. There's many ways to get involved: by engaging in research, becoming a land naturalist or land manager, doing an internship or student job, or attending events like these, assessing data, leading or contributing to events, or partnering with us on projects. You're very important in addressing the greatest environmental challenges in history. An engaged society is critical, and diversity is critical. Today, we're going to focus on water quality and how aquatic insects and other invertebrates play a part in water systems. We'll see how the adaptation of these specimens in local rivers and lakes can help to identify their water quality. We plan to explore the app called Aquatic Bugs to help us along the journey. This event is in the format of our local nature format, and we will combine a live presentation with an app exploration, which will be about an hour long. Some of you may have explored in local nature, which might include an area that you have access to during shelter in place, your back or front yard, the your window, maybe along the creek, and maybe some of you have taken pictures and brought them with you to the event today. So today we'll spend about 30 minutes hearing from Wendy St. And then 10 to 15 minutes exploring the app, and then coming back to discuss and ask questions until 3 p.m. During the presentation, Wendy may ask for your feedback, and you can answer her using the chat function by typing in your answer. The chat box is on the bottom of your screen in the black box, and there's a little button called chat where you can type in your questions. So hopefully you all are muted. So if you have not done so already, please mute yourself and use the chat box when you have questions. Or if you have comments like you cannot see or hear the presenter. Wendy will not be reading the chat until the end of her presentation, but I will be monitoring it. So feel free to type me or other people to address anything that you have. Okay, so now that I think everyone is here, I'm going to ask you all to type your names in the chat box right now. This will be our sign-in sheet for the day, since sometimes Zoom usernames are not the same as the names you registered under. So I really appreciate it if right now you could please all type in the chat your full name. Okay, let's see. Oh, I see. Oh, good job, everyone. Okay, I see your names. Thank you. Now I'm very happy to introduce Wendy St. a lecturer with the Department of Biology and Geography, Environment, and Planning at Sonoma State University. So welcome.
Great. Thank you so much, Nicolette. Can you hear me?
Yes.
Yes. Yes. Okay. So, yeah, as Nicolette said, I'm not going to be monitoring the chat during my talk, but if you have questions at any point, please feel free to type them in. And Nicolette, you can interrupt me. I'm happy to answer questions during the presentation. We don't have to wait till the end. So what I'm going to be talking about today is the importance of macroinvertebrates, particularly ones that live in freshwater systems. And we're also then going to play around a little bit with an app that's called Aqua Bugs that you can have on your phone. Hopefully everybody was able to download it. Again, all right. So this is a quote from somebody that I really respect. This is E.O. Wilson, who is a really well-known evolutionary biologist and entomologist and scientist who's done a lot of really cool things. One of the things that he studied extensively was ants. And this is what he says about invertebrates. If human beings were to disappear tomorrow, the world would go on with little change. But if invertebrates were to disappear, I doubt that the human species could last more than a few months. So what that speaks to is the importance of invertebrates in our ecosystems and in our lives. E.O. Wilson also has called invertebrates the little things that run the world. So invertebrates are animals that don't have backbones, right? That's pretty easy to figure out probably. So all of, insects, crustaceans, worms, snails, all those sorts of things would be considered invertebrates. And in fact, they comprise 96% of the known animal species. So of all the animals that we've described by science, only 96% of them are invertebrates. Now they have some really important functions. Let's go over just a few of their ecological importance. First of all, a lot of pollination is done by invertebrates, right? We associate bees and butterflies with this. Decomposition, something we might not think about a lot, but think about if everything that died just like laid around where it died, right? That would not be good. So insects are hugely important to that part of the nutrient cycle and breaking down dead things to their constituents so that they can those things can be reused in the future. It's very recycling, natural recycling. Invertebrates are really crucial to soil composition and the formation of nutrient-rich soils, like this earthworm. And then they're also foundational in many food webs. For example, here's a whale shark that's getting ready to eat a whole lot of krill, which are basically teeny tiny shrimp. It's amazing to me that an animal that size gains all that body mass by eating so many teeny things. So invertebrates are really important, and we want to give them the credit for that and really recognize that, because they get forgotten a lot, right? How many invertebrates are protected by the Endangered Species Act? I don't know the number off the top of my head, but it's not very many, because we kind of tend to forget about them. So let's look at then the other word that we're going to talk about today, which is benthic. So we're looking at benthic macroinvertebrates today. We often will see this abbreviated BMI. And so invertebrate, we know what that means. Macro just means something that's large enough to be seen with the naked eye, so that we don't need a microscope or a magnifying glass in order to view it. Now we're going to find that with a lot of the things we're looking at today, having magnification can be a really important tool to get in and see the features. But the animals themselves are visible just with the naked eye. And then benthic is a word that describes the part of the water column where these organisms are found. So we can talk about the benthic zone, and all that is it's the ecological region at the lowest level of any body of water. We can think about the benthic zone in the ocean, and maybe it's miles down and no light penetrates there, and that could be a benthic zone. But if we're talking about something like Copeland Creek, which is in the background behind me, most of the time that water is maybe 6 inches or a foot deep, and it still has a benthic zone. It's just that layer at the very bottom of the water, and then the substrate beneath it. So aquatic macroinvertebrates, again, just like with any macroinvertebrates, the diversity of these animals is vital to properly functioning ecosystems. We're going to find that they serve as food sources for lots of other organisms. Of course, we could think about fish, right? Eating insect larvae. This is a bird called a limpkin that's getting ready to munch down on a snail. So fish, amphibians, waterfowl all rely on aquatic macroinvertebrates. But there's also, maybe a little bit more surprising, is that there are a lot of terrestrial or non-aquatic animals that rely on these creatures as well for food. For example, this is a Wilson's warbler, and you can see in its mouth it's got some kind of a fly. Well, that fly, as we'll explore in a minute here, that fly does not live in the water, but it did as a juvenile. And that's where the aquatic part of that comes in, and how the stream environment is really crucial to their life cycle. And then crucial to all of the animals that rely on these insects and other types of organisms. They're not just all insects. Another thing that we can use aquatic invertebrates for is to use them as an indicator species. And I'm going to explain what that is in just a minute here. Really quickly, first, I just wanted to, go back a little bit to that, that we're used to when we're going to talk about mayflies, stoneflies, Dobson flies, lots of things that call themselves flies today, and none of them are going to have wings yet. So when we're thinking about them as being benthic macroinvertebrates, we're talking about the larval stage, or you'll hear this called the nymph stage. So when they're juvenile, these are animals that are fully aquatic. Eventually they will metamorphose and grow wings, and then they'll become flighted, and then they will leave the water. But we're going to be considering them at this time when they are aquatic. And then some of the things that we look at will just be aquatic throughout their whole lives as well. So what is an indicator species? A phrase that you may be familiar with, if you've ever heard of the canary in a coal mine, it was this idea that back when there was a lot of mining happening, sometimes there would be pockets of bad air, right, where there would be methane or whatever, some other kind of gas that would be harmful. And if people were down there mining and encountered that, it could kill them. So they would go down into the mines carrying a songbird, in a cage with them. And the idea was that canary was going to be an early warning signal for them. Kind of like now how we have fire alarms or smoke alarms in our house. It was the similar idea, but the idea was that if they encountered an area where the air was bad, the canary, because of its small size, would succumb more quickly to any bad effects. So if your canary dies, there's a problem with the air, and then all the people would need to leave as well. That's a little brutal. Anyways, but that's kind of what indicator species are kind of the canary in the coal mine, or we can look at it that way for a lot of ecosystems. So an indicator species is a species whose presence, absence, or relative abundance, right, how many of them are present, can reflect the environmental condition. So we can get information on the overall health of an ecosystem, and we can also get information on particular environmental conditions such as pollution, which we're going to talk about more in a second as well. An example of an indicator species, this is a Pacific, Pseudacris regula, I don't know what we're calling them these days, the Pacific chorus frog, Pacific tree frog, Sierra tree, maybe this is the Sierra tree frog now. The names have changed a lot, but what we can see here is that it has more legs than our typical, right? And this is a result of being infected by a parasite, a small worm that forms cysts on their where these legs bud out and causes them to grow these extra limbs. It's not great for the frogs. It's not a fitness advantage. But if we see a frog out in a habitat that has this kind of deformity, that's a way that we understand, oh, there's probably a parasite problem in this body of water. And we're going to look at the ways that we can also use these benthic macroinvertebrates as indicator species as well. So they can be really reliable environmental indicators because of a few characteristics. First of all, that they spend all or most of their lives in the water, right? So when we're looking at the nymphs, we're talking about, that's their juvenile phase. They will spend their whole juvenile phase in the water. They're easy to collect. Now, if any of you had a chance to go out and try that, you've probably already discovered this for yourselves, but they're easy to collect. We're going to talk about that protocol in a minute. And they differ, and this is the important part. They differ in their tolerance to pollution, which means we can look at the species that we find in any given area and use that to tell us something about what the conditions, what the pollution levels are like in the water where we found those organisms. And then they're also relatively easy to identify in the laboratory. The caveat to that is that they're relatively easy to identify to the major groups, and that's what we're going to focus on today. Trying to get these things down to species can be very much not easy, but we're not going to try to worry about that today. Now, I do have an asterisk by the word pollution, because I want to point out that when we're talking about pollution in this context, it's not necessarily what we think of as pollution when we use that word in everyday, just a sort of everyday conversation. So it can relate to the kind of pollutants like toxins, the factory up the road dumping out some sort of toxins, or the pesticide runoff from the local winery or whatever. It can mean that sort of thing. But also what we talk about with pollution are organic pollutants, so things like excess nitrogen, excess phosphorus, which those are things that exist in the ecosystem naturally, but if they're out of balance, it can cause a problem. And another thing that we would consider pollution in this context is we look at the amount of dissolved oxygen in the water. So dissolved oxygen is really important for the organisms that live in the water. So high levels of dissolved oxygen are good. Low levels are bad, and we can talk about low dissolved oxygen as being pollution in this context. And so we can see again how they differ in their tolerance to pollution. Some require really, really good, clean water with high levels of oxygen. Others, and then there's this gradient then of what kind of conditions they can tolerate. We'll look at that more in a minute as well. So how do we capture macroinvertebrates? First of all, just the caveat that anything that we're talking about here today, unless you have some sort of a collecting permit, you should not be collecting them to keep them. It's legal to scoop them up, examine them, photograph them, and then release them back again alive. If that's what you are doing, my understanding is that you do not require a scientific collecting permit, which would be issued by the Department of Fish and Game. But you will definitely, I would recommend that you want to check into whatever the regulations are where you are, just to make sure that you're not violating any laws. When you get into the water, though, to collect your macroinvertebrates, the easiest way is by dip netting, sifting, and straining. Basically, what you would do is we've got this woman pictured here. So she's got a big net. You could use a net, you could use a small net, you could use a colander. What would probably work really well is, there's like the big one for spaghetti. If you have one of those smaller ones where the mesh is a little bit finer, that would probably be fantastic. But basically, you situate yourself so that the water is running, like in this case, the water would be running from this direction through here, right? So that when you put your strainer in the water, the water from the flow of the river is pushing things into the strainer. Then you just set it down, like maybe in front of the rock or kind of near the bottom of the water, and then you agitate things. You can agitate the leaf litter or the substrate. You can rub some of the submerged rocks, and then doing this will dislodge these macroinvertebrates. They will get carried by the water flow into your strainer, and then yay, you can examine them. So these are some students of mine, and this was some collection that we did in Copeland Creek. Actually, probably exactly where this photograph behind me was taken. It might have even been the same day. Another thing that's really useful when you're going out to do this is to bring something else to view them in. I recommend something that's really light in color. Maybe you have a porcelain dish. The other thing that works really well are frisbees. Like if you have a light colored frisbee, because they're lightweight and they'd be great. But it's just something where you can take your specimens and really easily view them against a light colored background. Another thing that I would really recommend to you when you are, anytime you are doing this kind of collecting and going out and making observations in nature, is to keep notes about what you did. In this case, My students, obviously, we kept track of the date and where we were, the time of day, and then they made descriptions of all of the things that we found, because we weren't going to be able to identify them till we went back into the lab. And then they had some notes about the water speed, the habitat. Keeping notes like this is what elevates what you're doing from, hi, I'm like just kind of messing around and splashing in the water looking at some bugs, and oh no, actually now I am performing scientific investigation. It's literally just writing it down is what elevates it to being science in that way. Ideally then in a way that you could share with other people as well. But then we come to the identification. When I'm doing these sampling efforts on campus, we then will get everybody together in a room with a bunch of dissecting microscopes. So these are microscopes that aren't super high powered, but enough to give us, so for example, this is a photograph of one of our specimens. I probably took that with my iPhone through the microscope, although I do have a better camera as well. But what we do is we pull out books, we put all the specimens into little jars, and then we just sit down and identify them. Now, what we're going to focus on today, of course, is this app, this Aqua Bugs app, and there are other online tools as well. We chose this one today because it's really simple to use. It has a relatively small spectrum of like number of organisms in it, which is really great if you're just starting out and wanting to get your head around the major groups of insects. So, before we get into the app, really quick, I want to run us through some basic morphology of the kinds of animals that you're going to be finding. Here, I'm just looking at insects. Other things, if you find a snail, you know it's a snail, right? So let's look at some insect morphology. So we've got, because when you're going through the app, or when you're going through other references that you might access later, it may ask you, it may say, what are the number of cerci or whatever, right? And then you're like, oh, that's a tail. It might ask you how many gills they have, how many abdominal segments do they have. So you want to be able to identify that on this, so this is a mayfly, there's a head, there's a thorax, and there's an abdomen. That 3 body part structure is really typical of insects. And then there's going to be some number of legs. In this case, we can see, well, we'll talk about legs in a second. So those are the 3 segments. You may be seeing structures like gills. You may be looking for antennae, right? You may be wanting to count the number of abdominal sections. Here's another example. This is a caddisfly larva. So some of the things you may look at is the shape of the body. This is a comma-shaped body rather than straight. Again, a head, a thorax, and an abdomen. Looking at the characteristics of the tail is going to be important. Looking to see whether, again, abdominal gills can be important. And then here, finally, we have a Dobson fly larvae. And this is the one that I wanted to point this one out, because this is something that when we're talking about some of these juvenile flying insects, something that can trip people up is that insects have 6 legs, right? That's a feature of insects. And then you look at this and you say, oh, that has a whole lot of legs. It must be something else. But only 6 of these are true legs. So here on the mayfly, it's kind of easy to see the legs. There's 6 of them. They're jointed. These are true legs. On the Dobson fly, it also has 3 sets of true jointed legs. And the rest of these are what we call fleshy appendages or false legs. So at this stage, they have these false legs. They're not true legs. So just be looking for that when we're working with the app. Okay. So let me walk you through. There's 3 ways really to approach using Aqua Bugs for identification. When you open up the app, and feel free to kind of play along with this at this point if you want to, you can start by looking at legs, by looking at body, or looking at tail. So let's go through an example with legs. So I've got this is something that I pulled out of Copeland Creek. We can look at the morphology of it just really superficially, excuse me. What I really mostly notice is lots of legs, and they do look like true legs, right? These look like they are all jointed appendages, probably true legs, not those false ones like the Dobson fly had. So when I click then on, what am I doing? Legs. A screen is going to come up, and it's going to make me ask me to make a choice. So what we would call this kind of a system, it's called a dichotomous key, where at every point you're asked to pick, well, is it this or this? And depending on that answer, you'll then be asked to make another choice. And each choice is pushing you nearer to the correct answer. So here, when I'm looking at how many legs it has, I'm pretty sure that I'm going to pick it has many legs. When I do that, this screen pops up. Every time you do something with legs, this screen will pop up. Because if you say that it has a lot of legs, it's going to say, are you sure it's not one of these things with false legs? We're sure. In this case, that it has many legs. Let's move on. So now, does it have a worm-like body with caterpillar legs? Or does it have a lot of legs and it's not worm-like? This one looks more like this to me. And now this is going to pull up, does it look like a lobster or a shrimp? Or does it have this body wider than it is tall, and etc.? This one might be tricky because superficially this almost looks a little bit like this. Until I realize that it resembles a lobster or a shrimp, oh, that definitely looks more like a shrimp to me. So I'm going to pick that one. And now it's going to ask me, does it look like a lobster or does it look like a shrimp with longer legs? Definitely looks like that second one. And then it's going to ask me another. Now, you don't always have this many questions, this many steps. But here, does it have this shrimp-like body or does this fan-like tail? We're going to go with that top option. And it's going to tell me that my organism is a scud. Now, that is correct. And if you look at it, yeah, that's exactly what my organism looks like. So let's go through another option, another example. So here we have something that I want to identify, and let's use body type. So I start out and it's going to ask me if it has one or more shells or if it has no shells. No shells. Now it's going to ask me how many legs does it have. Now again, I could maybe go with this many legs option. Even if I say 6 legs, this screen will pop up and now I need to make that decision. Oh, I think this is one of those ones that has the false legs. So let's try that. Now it's asking me the body longer than it is wide, or is it oval and disc shaped? I'll pick the right option. Now it's going to ask me how many tails it has. Sometimes this is really obvious. Sometimes it's not. This is one of the not obvious ones. It might look like it has these little 2 tails here, but really those are other structures, and we're going to go ahead and call this one having no tail. So we're selecting no tail. Now it's going to ask me, is it comma shaped or is it straight with the legs out to the side? We're going to go with straight legs out to the side. Now, does it look like a beetle or does it look, something else? The abdomen is the same with fleshy abdomen. Definitely that second option. And now it's going to ask me if it has gills on the under tufts of the abdomen or no gills. So let's go back and look at our specimen. If we blow that up a little bit, we can see, oh, yeah, there are definitely these little fluffy structures here that we're going to call gills. I select that option, and it's going to bring me to an answer, which is the Dobson fly larva, which is correct. Now, what if I had chosen the other option? What if I had not really realized that there were gills and I'd selected this one? I would have had some different steps, and then it would have taken me here to the whirligig beetle larva, which is wrong. If that happens to you and you feel like that's not the right bug, what you can do is click this button, Not My Bug, and it will pull up some similar species, and hopefully the species that you have there will be one of those that pops up. Boom. Okay, last one. Let's try to identify something by the tail. Here, again, this is one that has a really obvious 2 tails. Let's go with 2 tails. When I click 2 tails, oh, sorry, that was already up there. When I click 2 tails, it's giving me 2 options. That it has hair-like tails and no gills on the sides of the abdomen, or that it has webbed tails and gills. When I look at this, I'm not seeing any gills. Let's select that one, and boom. Oh, that was easy. It's telling me that this is a stonefly, which indeed it is. Okay, the other way that you can try to find things in this app is by using the list all function, which shows you sorry about that, shows you the entire list of all the species that are in the program. It's not a huge list of species, but this is a way that you could use that as well. So what I want you to do now is grab up your app, and I am going to look at the chat now. Pull up your app, and I'm going to show you 5 specimens, and I want you to try to identify them. So as soon as somebody has identified them, go ahead and type what you think it is into the chat. And then I'm going to move through these. I'm going to give us probably about 7 minutes to do this. And then we're going to talk about pollution tolerance. So open up the app and yay, Patty, well done. So if people don't want to don't look at the chat yet, see if another few people want to get there, but Patty has definitely gotten to the right place. And then you can look at that and you can understand why it's called that actually. So yeah, if somebody else wants to tell me what they think this is, I want to make sure everybody has time. Carolyn, well done. That's okay. No. Well done. Is that Pence? I'm not wearing my glasses. Well done. Okay, good. So I think we're off to a good start here, right? People are able to do this. So let's start with let's do another one. Oops. There we go. Okay. How about this one? So of course, what you're going to think about, right, is where are you going to start? You know, body, legs, or tail. It kind of depends on the specimen. I know where I'd start with this one. I'd start with the tails. And then of course you can use the little, I've highlighted some of the features that you would be able to see if you were looking at them under a microscope. So I've highlighted some of those. So Patty, you're close. You're on the right track, but yes, Christina, yes. Yeah. Now, what are the things that make this a mayfly and not a stonefly? If you looked at that, and this is where it's tricky, and this is kind of the reason I put this one in here, because mostly when we think about 2 tails, we assume it's a stonefly. Mayflies have 3 tails, but this one has 2 tails, but it also has these gills. So if you looked at that 2-tail slide, you would have said, oh, those abdominal gills, this is one of those 2-tailed mayflies. Yeah, exactly. And that's why, you know, it's pretty easy to get to like the big scale groups of these, but some of them, there are some of the differences that are going to let you help to determine them can be really, really nitpicky. So this is just some practice, like looking at what those kind of characters are. Okay, number 3, see if you can find out who this is. I always look at this and think this is the face, but it's not. That's actually the tail end of the body. This is the head. We've got some antennae here. Yeah. This is an animal that has, I know, it just looks like the face. This is an animal that has terrestrial relatives that you're probably really familiar with, which I won't say until somebody's gotten what it is. Because it looks like a face and he's got like a little pinch your hands. Hi, here's my pinch your hands. Instead I have a pinch your booty. Okay. Oh, this one's taking a little longer. I think there's kind of a lot of steps to this one. And then don't get tripped up when there's going to be a thing that's going to ask you if it's wider than it is tall. You can't see that in the thing. Yes, well done, Christina and Carolyn. Yes. So this is an aquatic sow bug. A more sort of technical term for this would be an isopod. Oh, I guess I could have said the scud at the beginning. That would be an amphipod. That's the sort of, and these are crustaceans. So this that we're looking at on the screen here is closely related to pill bugs or roly-polies that we could find walking around in our garden, right? So, okay, well done. People are rocking through this. Okay, how about this one? This is maybe a little easier in terms of what it is in general. Yay, Patty, I'm glad that you're enjoying this. I know it is fun. It is fun. That's one of the reasons that I like to do this project. So every year I will take some students from the entomology class out into the creek, also from my restoration biology class, and we collect up specimens and then we go back and identify them. And it is fun, and sometimes it's frustrating, but it's a great project. And it's something that you can pretty easily do in your own neighborhoods as well. This one's taking a little longer because you're going to get to a screen. Yes, thank you, Christina. You're going to get to a screen that's like, this looks right. And then you click that, and the next screen is like, wait, what? And then you're going to get back on track again. So yes, Christina has gotten, yes, Patty, well done. Awesome. So, okay, perfect. I'm going to go to the next one because the next one's a little bit tricky and I want to make sure we don't run out of time. Yes, this is a riffle beetle. Okay, last one. Now, for those of you who maybe know something about these, this is maybe the easiest one. Going through the app, this is going to be the hardest one that we've done so far. So just notice that it has 2 armored plates on the back of the head. It does have 6 legs, and we're going to call this a hooked tail. And it's living in a little house. Yes, Patty. Now, did you use the app or did you know? Okay, yeah. The house is the dead giveaway, right? Yeah, going through the app on this one it takes a little bit longer. So I'm gonna give it another minute here and see if there's anybody. If you get through this and you find this in the app, type that into the chat and then we'll move on.
Wendy, by the way, it is 2:40 right now.
Awesome. Thank you. Okay, so we'll move on. Please feel free to keep kind of playing around with this one, but I want to make sure we have time for the pollution tolerance. So these are our specimens, right? So what we had were a water penny, a mayfly, an isopod or aquatic sow bug, a riffle beetle adult, and then this last one was a caddisfly. And what's funny is really once you get to know them, the minute you pull something out and it has that little, it's built itself that little house, it's a caddisfly. It's the easiest thing to identify. It's a lot of steps through the app though. Okay, so let's then wrap this up by talking about how we use these as an indicator of water quality. So what we're talking about is stress tolerance. Basically, what kind of conditions each of these different species and groups need in order to survive. Now, we can talk about natural causes of stress or things that make changes in an environment, erosion and sedimentation, forest fires that can change the chemical elements that are being washed into the creek, floods, landslides, and then of course, we're also going to have, we can think about anthropogenic reasons. So things that humans have done that impact environments, pollution, when we're removing water by irrigation, when we're building dams, deforestation, removing riparian vegetation, all of those kind of things can, another really big one is like runoff of fertilizers into water that can really change the quality of the water. So what we want to do is how can we use these invertebrates to get an idea of what the water quality is like. So these invertebrates they vary in their ability to cope with environmental stress. And so we can do something called biomonitoring, which is going to help us identify, whether the aquatic environment, are most of the organisms there, like, do they need really clean water? Or do most of the organisms living there just really don't care that much about the water conditions and they can live in bad conditions? And that's going to tell us something about our water quality. So today I'm going to break it down into 4 groups. When you look at other references, you may see different numbers. Sometimes they break it down to 3. Sometimes they break it down to 10. But the idea is the same. Which ones need what level of cleanness or high quality water? Group 1, these are going to be very sensitive to pollutants and they need a lot of dissolved oxygen. So some of our mayflies, our stoneflies, our Dobson fly, those are the ones that I see regularly around here. Group 2. These are semi-sensitive to pollutants, and they require moderate to high levels of dissolved oxygen. In other words, they can tolerate slightly worse conditions. Our riffle beetles, water penny, many of our mayflies, our dragonflies and damselflies, crayfish, caddisflies. Those are some of the group 2 taxa. Group 3, these are ones that are pretty tolerant of pollutants, and they only require moderate oxygen levels, black flies, the green midge or non-red midge flies, and then our amphipod or scud. And this is just a sample. There's more organisms that we could look at. And then finally we have our group 4. These are the ones that can live in poor quality water, so they don't need a lot of oxygen. They're very tolerant of pollutants. Leeches, red midge flies, aquatic worms, our little isopod, a lot of the snails. Another thing that we will also often think about as being group 4 are some of the things like these water boatmen or water striders. Now, these are only, the reason that these can tolerate bad water conditions is because they're not living in the water in the same way, right? The water striders are on the surface. The water boatmen are not, like they're not as integrated into the water, and so they're not affected by water quality. The presence of them doesn't mean bad water quality. You'll find them in all types of water. So how would we use this to assess water quality? And we're going to power through this. I think I can do this in the next 2 minutes, and then we'll be right on time. So what you would do is you would want to catalog the species that you found. So let's look at what we identified today, all of which are things that I regularly find in Copeland Creek on campus. We found a Dobson fly, a mayfly, and a stonefly that would be in that very intolerant or require really, really good water condition. The moderately intolerant, we found a caddisfly, a riffle beetle, and a water penny. In our fairly tolerant group, we found an amphipod. And then in our very tolerant, we found that little isopod. So how are we going to use these to calculate or to assess water quality? First of all, we just tally up how many from each group we found, and then we add that up. So we get a total of different type of organisms that we found. Now we need to come up with a scheme of weighting them, right? So that we're now including in the fact that some of them require better conditions, some of them require worse conditions. So what we're going to do, we're going to multiply these numbers by this weighting factor, and all the weighting factor is the number of the group. So if they're very intolerant, you multiply it by 1. If they're moderately intolerant, we're going to multiply that by 2. So we get the 3 times 2 is going to be 6. Our group 3 multiply by 3, and our group 4 multiply by 4. So if you see what I did there, right? So we're putting a weighting factor onto this. Now I can total up those numbers. So now we can see that our weighted scores is a 16. Our actual total of organisms is 8. Now we're just going to divide one of those numbers. We're going to divide our number of organisms into our weighted score, and that's what gives us our pollution tolerance score. Again, we did this on a thing where there's 4 groups. You could do the same thing using a different scale, like a 1 to 10 or a 0 to 10, and it'd be the same idea. So now I take that score, I look at my pollution tolerance chart here that's based on this 1 to 4 system, and I can recognize that my water quality in Copeland Creek is excellent, because what we have here is more of an emphasis on species that are very sensitive to bad water, species that require high levels of dissolved oxygen. If those are present, then you know the water quality is good, and that's how we're going to use this. Now, really quick, I just want to throw at you some additional resources. You're going to find that the Aqua Bugs app is a great way to get started, but if you really get into this, almost immediately you're going to want to be able to go deeper into some other resources. So these are a few things that are available for you online. I can figure out a way to make these, that these links can be made available through the I'm sure that Carrie can help me get them online somewhere. This is a really good one, this California Digital Reference. You'll start here with all of these different groups, and then they have a profile on every single all of the different groups. It's really in depth and they've got really excellent photos. So this is where I would send you next, is one of these others that are going to be a little bit bigger than the app. If you really get into this, check out this digital reference collection. And that is what I have for you today. So oh, great. Let's see. Patty, I'm glad to hear that. Thank you. That's really good. And Patty, also, I'm going to reach out to you soon because there's something else that I wanted to ask you about with the Sonoma Mountain thing that you did. So this method seems like it would be influenced by survey effort. Is there a duration of time? Does it matter if you find many of some taxa and a few of others? Those are great questions. So yes, the method can be influenced by survey effort. Now, to some degree, if you go to a body of water and you're scooping out some stuff and you're finding a bunch of mayflies and stoneflies, it doesn't really matter how much time or whatever you spent, because you're looking at insects here who cannot exist there if the water quality isn't good. So that's telling you something. But we definitely can put onto this some protocols that's gonna make our data better. It's gonna make our data better and more useful to compare to other things. So the protocol that I use, which is pretty loosey-goosey in terms of protocols, is that we go out and we do a 45-minute sampling effort in any location that we're using, and then everything that we've collected, then we will categorize them. In terms of the numbers of the few, how many are present of each taxa, I think that you could include that in your calculations. I haven't ever done that. And full disclosure, I'm not an entomologist. I'm an ecologist who ended up working on this project, and I really love it. But there are definitely other people who are a lot more, have a lot more experience with the actual organisms than I do. But we definitely do keep track of abundance, but I've never used that in my water quality measurements. Although I think that you certainly could. That's a really great question that I can't really give you a solid answer on. But thank you, Kate. Great questions. Any other questions? Okay, cool app. How often does the Aqua Bugs provide the accurate answer to students? And how often are incorrect Okay, so that's a great question. So in my personal experience with this, I'm going to say giving percentages is a little dodgy because this is anecdotal off the top of my head. I feel like when I have correctly identified the traits, I'm going to say that I get the right answer like 95% of the time. If I haven't really stopped to look, oh, does it have abdominal gills? Oh, how many plates behind the head? Then that percentage goes down. And most of the time it shows up on the similar species, but definitely not always. That was a problem that I had, for example, with the caddisflies, that a lot of times I would go through caddisflies and I would go through so many things and never get to caddisfly. So it's definitely, you need to have some level of knowledge about these organisms in order to use the app. But that's why I like that list all function, because it has such a limited number of species in it that if your species is there and you look at the list, you can actually just scroll through until you find the picture. Now, maybe you found something that's not in the app. That could definitely happen. Although I'm trying to think of I think most of the things I regularly find in the stream are actually included in the app, but then you might want to go to some other, and there's a lot of online things where you could look at, there's dichotomous keys and things online. There's a lot of really good resources online for this, so it's a good question. The app is not fail-safe by any means. Okay, so Carolyn is asking, what tools do I recommend for viewing structures if you don't have a special microscope? Great question. The thing that's nice about these is that they are big enough that if you were to get just a magnifying glass, or maybe like one of the things that's kind of cool is, I want to call it like a jeweler's loupe, but it's not. It's actually for viewing. And so you can get either like little jars that have a magnifying kind of dome on the top, or something like that, or one of like, it's a like a little cup that's turned upside down, and the top of it again is a dome that gives magnification, and you could just set it down over the top of your specimens. you don't need a lot of magnification to get at many of the structures. Now, if you're trying to get to species identification, and it might add, and then some of those you might need to look at very, very small structures, then you'd want a dissecting microscope. But you're going to be able to find most of these features, even with just the limited magnification you could get with like a regular magnifying glass, or a little bug identification kit thing that would cost you, $5 or $10. Oh, speaking of one thing that I did want to mention, they're calling this app Aqua Bug. Remember that not all of the things that we're looking at here are bugs. From a scientific standpoint, the word bug, ooh, am I frozen? The scientific standpoint, the word bug only refers to this one group of insects, the Hemiptera, the true bugs. Everything else, when I say bug, I'm using it in a colloquial, casual way. Okay, so Patty wants to go scooping and sifting now. Do it, and then take some pictures and send me some pictures. I would love to see what you find in your local waterway. So Carrie is sharing that she usually gets to the correct answer because otherwise my organism looks wrong compared to the picture on Aqua Bugs. And she is suggesting reading glasses as a way. Oh yeah, for sure. Reading glasses, you could really use reading glasses to be able to magnify them. They don't require a lot of magnification, so that part is pretty easy. So if we send pictures back, where do we send it? So my email address is, and we could definitely make this available somewhere, but it's stjohnw@sonoma.edu. Or if you sent them to the Center for Environmental Inquiry, just referencing my name, they'll get to the right place. And then I can even maybe make a way to post some of them online in association with this. So, Julie, could you please briefly discuss the best method to get the greatest quantity and diversity of aquatic insects in, say, Upper Copeland Creek? Okay, so these insects are going to be distributed. I haven't really noticed a lot of differences by going different areas. So what you want is you definitely want to go into areas where there's flowing water, where the water is stagnant. You still will find some things, but I'm not sure you're going to find the same community. Or maybe you sample in a more of a stagnant part and you sample in a part that's running. But the effective thing to do there is to get your thing down so that it's actually maybe in this, even a little bit in the substrate, and then really just agitate leaf litter, the substrate, and then the rubbing rocks thing is a really good strategy. In fact, sometimes you can even you can look at the rocks and you can see things attached, like you can see a water penny attached. You can see mayflies or stoneflies. So just rub them and let everything get carried into the thing. Doing this sampling it's ridiculously easy. I have found it ridiculously easy to get samples, and we almost always have more samples than we can comfortably identify in sort of a reasonable amount of time before people just get really tired of it. So I haven't ever looked into any ways to try to increase my sampling, because the sampling we've always gotten a really good diversity and a lot of samples that way. But I would just suggest that going to a couple of different locations in your waterway is going to pick up the greatest amount of diversity.
Okay.
And then, yeah. Huh?
I'm going to send the outro, and then if you have a few minutes after to answer the remaining questions. Does that sound good?
Sure. Can I just really quick answer these last two questions that are here?
Sure.
Okay,
Yeah.
so Christina asked, what if you find an area that potentially has significant pollution? Is there a way to bring attention to it? That's a really, really great question. So it depends on if it's who owns the land. That might be something you could reach out to the water agency with. Let me think on that, and I can try to come up with a better answer to that, and maybe put some resources up online somewhere, because that's a great question. Now, if it's your land, then you could look into maybe some ways of mitigating the water. That's a whole other topic, but that's a really good question. And then Jessica is taking a picture with your phone and zooming into magnify. That also works really well. I've gotten some amazing photos with my iPhone. So yeah, that's another great way that you could get a closer look at some of these. Okay. Thank you, Nicolette.
Okay. Thank you. Okay, I'm just going to wrap up really quickly so we can end at exactly 3 o'clock. So thank you again, everyone, for attending. I hope you all learned a lot and feel empowered to use, sorry. Okay. Hello. Okay. I hope you all feel empowered to use these skills a little bit more and have some fun with it. We have some great resources to do some further learning. Again, please feel free to email me or Carrie or Wendy after we sign off, and we'll all try to reply. This is one of our citizen science local nature style events each Friday at 2 p.m. through mid-May, and one of over 24 events the center has planned through June. And more events and learning opportunities are being added all the time. So check out our website, cei.sonoma.edu, and to pre-register. Feel free to spread the word, even to those who don't live in the North Bay, because with our virtual events, anyone can attend from anywhere. Some of the specific events that are coming up that may be of interest to you all include tomorrow, Saturday, May 16th at 9 a.m. There is a citizen science event, which is backyard wildlife cameras, led by Ben Bravo and Andrew Balistracci, who both have experience using wildlife cameras on our preserves. They will talk about the types of cameras and how to optimize the use of the cameras so you can discover the wildlife in your backyard. Next Friday, May 22nd at 10 a.m., there's a local nature event called Backyard Photography, led by Rick and Jeray Waller, who are the founders of Heartwork Accessible Nature Photography. Come learn creative techniques to discover your backyard in a fun new way. The next Friday after that is May 29th at 2 p.m., which is Learn with a Naturalist, Storytelling in Nature, led by Sonoma State naturalist John Lynch. Come listen to John's riveting stories and maybe create a joint story together. So thank you, everyone, and stay safe, and we hope to see you soon.
Great. Really quick, I just wanted to point out that in the chat, I put the URL to my website. It's teacuprex.com. That's my personal science blog. You're also welcome to reach out to me there. I haven't been updating it lately, but I intend to start updating it some more. And there's a bunch of stuff there, so you could get in touch with me there as well.